So during today's lesson, we're going to be looking a little bit more at plant responses to the environment and specifically at plant hormones. Now, I'm going to give you a moment to just think about how many plant hormones you might be able to name. Are you familiar with any already? So one of the most common plant hormones perhaps is auxin, involved in sort of the growing of the plant, flowering and fruit development. We'll also look at gibberellins and ethene, as well as ABA and the action of cytokines as well in plants. Now, there's loads of similarities between plant hormones and animal hormones. So in both cases, for example, we see that they're transported through transport systems to target cells. They are responsible for setting off a whole range of different reactions in cells. They will bind to receptors in plasma membranes or on the cytoplasm. So they act in a very similar way to animal hormones. But we also see quite a few differences in plant hormones as well. So, for example, a single plant hormone might have a very different effect on different parts of the plant when it's in different concentrations, etc. We also know that animal hormones, for example, are produced in glands, whereas for plants they obviously don't have glands. Instead, they tend to be created or produced in tissues known as meristem. Now, lots of the experiments that are done on plants, specifically looking at the action of hormones, are done on this type of plant here, Coleoptiles. The coleoptile has a few really important areas that you need to be aware of. So, for example, towards the top, you can see at the apical meristem, there are small undifferentiated cells there that are constantly dividing. Now, some of those new cells are going to grow longer so that the length of the shoot steadily increases. And that's essentially how the plant grows. Now, this coleoptile is going to be particularly important as we look at the experiments done to show the action of auxin. Over the years, way back to kind of Darwin back in 1880, experiments have been done on coleoptiles and looking at the action of auxin in them. Now, Darwin began by looking at or just observing intact coleoptiles and seeing that they bend towards light or a light source. They are positively phototropic. Now, what Darwin did back in 1880 was he actually cut off the tip of the coleoptile, and he noticed that when you expose the rest of that shoot to light, the coleoptile will no longer bend in response to the light. Now, in a third little experiment, he covered the tip of the coleoptile so that it was shaded. And again, he observed that it would not bend in response to light. Now, based on these experiments, Darwin made a series of conclusions. He concluded that the growth stimulus must be produced in the tip of the coleoptile, and that the growth stimulus is transmitted to the zone of elongation, as he referred to it. So it goes down from the top to further down the stem. And the cells on the shaded side of the coleoptile will elongate more than the cells on the other side. And the way that he showed that was in that first experiment on the left-hand side there, that depending on the direction of the light source, that will dictate which way the coleoptile will bend. Now, a little later on, we're now into 1913, Boyce and Jensen did a series of experiments to help further our understanding of the action of auxin. Now, Boyce and Jensen used two different materials, one which was permeable to water and the other that was impermeable to water. So first of all, he used mica, which is not permeable, and he placed a sheet of mica, he inserted it through the shaded half of the coleoptile, and this stopped the curvature response, and that's what's being shown in that first diagram. In the second image, you can see that Boyce and Jensen inserted a sheet of mica through the illuminated side, and this allowed the curvature response. Now, in the second set of experiments, he used a sheet of agar or gelatin, which is permeable to water. And he cut off the tip of the coleoptile. He put the tip onto the block of gelatin and then exposed it to a light source from a single direction. And again, there we saw the normal curvature response to light. Now, based on these experiments, Boyce and Jensen concluded that materials which are not permeable to water, such as mica, can stop the curvature response in some circumstances, whilst materials that are permeable to water, such as gelatin, do not interfere with this curvature response. Finally, you need to be aware of the experiments that were done by Wendt. These followed on by those of Boyce and Jensen. This is some kind of like 10, 15 years later now. And Wendt placed the coleoptile tips on top of agar. By doing so, he drew the growth hormone, which we now know to be auxin, out of the tips and into the agar. He then placed the agar blocks onto the coleoptile and observed what happened. Now, based on his observations, he saw that the coleoptile actually bent, it curved when it was exposed to the auxin, as the auxin seemed to be causing some form of elongation to the cells on the side of the plant that it had placed that agar block onto. So his conclusions were that the angle of curvature is related to the number of tips used, i.e. the concentration of auxin in the block. The result fits a hypothesis that the curvature response is due to chemicals which move from the tip and into the area of cell growth as it affects cell elongation. Now, we're now going to look at essentially how auxins work, how they cause this cell elongation. Phototropins are responsible for absorbing blue light. When they absorb blue light, they become phosphorylated. This causes transporter proteins, known as PIN proteins, to increase the efflux of auxin, and that is shown in the first diagram. So in the first diagram on the left-hand side, you can see how we've got an efflux of auxin through the PIN proteins. Now, auxin is responsible for promoting the active transport of hydrogen ions into the cell wall. When this happens, we have a lowering of the cell wall's pH, and this is shown in that sort of central part of the diagram there. Expansins, which are wall loosening enzymes, work best at low pHs. They break bonds within the cellulose so that the walls become less rigid. The cells take in more water as a result and expand, elongating, resulting in the lengthening of the shoot. Now, this diagram shows this again. We see the auxins being synthesized in the meristem cells, so that's at the very, very top of the coleoptile. The auxins diffuse away from the tip, binding to receptors. That binding causes a lowering of the pH as we see the active transport of hydrogen ions into the cell wall. And the low pH keeps the cell wall flexible. The way that it does that is through these expansins, the wall loosening enzymes that work best at this low pH. And ultimately, this enables the cells to take in more water and elongate. Now, it's important that you're able to interpret graphs like this. Now, That's IAA is the abbreviated form of auxin. It's the most common auxin that we look at. And what you can see here is that over time, as auxin levels increase, we see a decrease in pH. Now, again, what we have just learnt is that when we see an efflux of auxin, auxin promotes the active transport of hydrogen ions into the cell wall, lowering the cell wall pH, which is why, as we see an increase in auxin, we see a decrease in pH. Now, the effect that auxin has differs in the route to the stem. So in the root, auxin is actually responsible for inhibiting cell growth. In this case, what we see is that the lower cells will become shorter, and this enables a turning down of the roots down away from light. In comparison, in the stem, auxin stimulates cell growth, as we've just seen. As it stimulates cell growth, this enables the stem to move or grow towards the light. The stem is positively phototrophic, whilst the root is negatively phototrophic. The next process we're going to look at is a process known as apical dominance. Now, most plant shoots have buds on either side of them called lateral buds. The presence and growth of the apex of the shoot inhibits the sideways growth from these lateral buds. That process, that phenomenon, is known as apical dominance. Now, it's quite easy to demonstrate, and many gardeners will make frequent use of this apical dominance. If you want a plant to stop growing upwards and instead you want it to start branching out sideways, all you need to do is simply cut off the tip of the main shoot. This removes the apical dominance and enables the lateral buds to grow. Apical dominance is shown in our Christmas trees. That's a good example of where we see it. And the lack of apical dominance is shown in plants like the whooping larch or the weeping larch, as you don't have that inhibition happening from the active apical bud. So the lateral buds don't remain dormant, instead they're active, which is why you see the shape of that weeping larch. Now, the exact way in which the inhibition of the lateral buds is caused has not actually yet been worked out. However, we do know it involves auxin. Auxin's constantly being made by cells at the tip of the shoot. It's then transported down from cell to cell. The auxin accumulates in the nodes beside the lateral buds, and somehow its presence inhibits their activity. Exactly how that occurs is not quite known, but there have been a series of experiments that have been done to help us understand. So in the first experiment that you need to know about, you can cut off the tip of two shoots and apply synthetic auxin to one of them. This one will continue to show apical dominance and the side shoots will not grow, whereas the other one, which has not been exposed to that synthetic auxin, will branch out sideways. A second experiment that can be done is if a growing shoot is tipped upside down, apical dominance is prevented and the lateral buds will start to grow out sideways. This can be explained by the fact that auxin is not transported upwards against gravity, but only downwards. So in the sort of upside down shoot, the auxin produced in the apical meristem doesn't reach the lateral buds and therefore it can't affect them.